Good morning, everybody. Thank you so much for taking the time to join us today. I'm so excited to be here today and share with you Purolite's offering in the bioprocess resin space. Today, I'd like to talk to you about our unique and novel manufacturing technique, two of our Protein A products, and I'd like to share the stage with one of our customers who will share a case study using our Jetaday 50 high pH resin for the purification of a bispecific antibody. My name is Tony Thomas, and I'm a field application specialist here at Purolite. I support the Western geography of the United States, where I help customers through process development, scale up, as well as optimization of existing processes. Also there to support with any other ancillary questions that come up, maybe around regulatory or quality or stability, anything you can really think of. And today, Kayla will be joining me from MabPlex, where she is a senior engineer within the downstream process development group. And she will be speaking to the purification of a bispecific using Jetaday 50 high pH, as mentioned earlier. So this is the structure of today's talk. I'd like to talk a little bit about Purolite, and then I'd like to go into our unique manufacturing process, which we refer to as jetting technology. After which, I'll talk about Jetaday 50 high pH, our latest Protein A product. It is truly unique in the sense that there is nothing else like it on the market. It allows for the elution of FC-containing species at higher pH. And lastly, Kayla will join and talk about the MabPlex case study, where they use Jetaday 50 high pH to purify several acid label bispecific antibodies successfully, to mention. So Purolite, who are we? Purolite was acquired by Ecolab in the fall of 2021. Purolite is a resin company first and foremost. We provide resins for the purification of monoclonal antibodies as well as recombinant proteins. We have ion exchangers, we have protein A affinity ligands, and with that we also offer a great support service with our field application services and our commercial teams. Ecolab as a whole operates in many different areas, too many to list, but for example, water purification, mining of gold, like treating nuclear waste. There's so many different things, but for the purposes of this discussion, I'd like to highlight that Ecolab also provides environmental cleaning and disinfection services and products, production equipment cleaning, as well as automated decontamination services. Purolite entered the bioprocessing space roughly in 2015. That's when we introduced our resins to the life sciences market for purification of molecules. And in that time, we have established ourselves as a market leader in the protein A and ion exchange space. We are embedded in over a dozen commercial processes and work with many of the world's largest therapeutics companies and CDMOs. We are found in hundreds of clinical trials and are able to supply thousands of liters of resin without issue. So to really understand our value and our uniqueness and our advantage in the marketplace, we have to take a step back and look at how beads have been traditionally produced. Resin, the backbone of purification using chromatography. And if you really think about it, there really hasn't been any serious innovation since resin was first manufactured in the 1960s. This is really the first innovation of its kind in that time. So let's look at the way we traditionally produce resins. Resin is made up of agarose, which is typically extracted from red seaweed. You find this off the coast of the beach, and you take this and you extract the agarose and you put it into this stirred tank reactor. During an emulsification process using organic solvents, you have droplet formation. The rate of the impeller as well as other variables kind of control the distribution of that particle size. Once you have your particles, you cross-link them with epichlorohydrin. That's what gives a bead its pressure flow properties. after which you screen, because in this emulsified process, you have a wide particle size distribution, which means you have to remove the fine particles as well as the coarse particles. And this is a very time-consuming step. It accounts for roughly 50% of the total processing time, and the ends of the fines and the coarse particles end up being disposed of. You basically waste everything that you don't use within your specification for particle size distribution. Once you have your target particle size distribution, you can then functionalize the bead with the ligand of your choice and package it for use in the formats that our customers need. The main takeaways here are you consume a lot of organic solvents, you consume a lot of water, there's a lot of waste with the screening step, as well as a lot of time. It takes extensive time to manufacture, and it consumes a large footprint. So that leads me to jetting technology. This is Purolite's unique patented manufacturing process for agarose beads. And you'll find that it's very different to the traditional emulsification techniques for agarose base matrix production. Here you can see two reactors. One contains agarose and the other contains heated mineral oil. And these two solutions are pumped into the center. In the center, you'll find this blue membrane can. This blue membrane can has about 20,000 laser cut holes. And these holes dictate how large the particles really are. As this can is being oscillated, the variables you see on the left of your screen, such as the design of the nozzle, the flow velocity, the viscosity, these all contribute to what comes out of that can. And what comes out is a pretty uniform-like distribution. So there is no screening step here. You don't have to spend all that time removing the coarse and the fine particles, and you have a tighter distribution, after which you follow the same steps as you would in an emulsified process. Takeaways here are this process is semi-continuous. You have a very small footprint. That can in the middle is about the size of a soda can, maybe a little bit larger. Also, you don't consume as many organic solvents or water. So we can manufacture these batches in roughly half the time it would take for an emulsified batch, which really adds value for our customers. The footprint of this process is also much smaller. We are able to translate this to facilities relatively quickly. So what does that look like? If you look at this slide, there are two graphs, one at the top and one at the bottom. The top is a market-leading protein A. That's the particle size distribution that you're looking at. And at the bottom is our jetted A50 protein A. You can see there's quite a distinct difference here. Our distribution tightly wraps around the average particle size diameter of 50 micron, whereas on the top, you see there's a wide range of particles. And at the top right of these graphs, you can find a value called the uniformity coefficient. And for our jetted process, it's less than 1.3. But for the emulsified technique, you're closer to 2. What this value really means is the closer you are to 1, the more uniform you are. And this is truly a byproduct of our jetted manufacturing process. But what does that actually mean in terms of purification? How can it impact your workflow? If you think about a column, theoretically, you pack it. And on the left, you have a pack that was done using an emulsified batch. And on the right, you have one using a jetted batch. And these are just representations. But this is what typically happens in that column. You have molecules that enter the column at the same time. However, if you have a wide distribution of particles, there are multiple paths that you can take through that column. And with that, you start eluting at different points in time. So what you see there on the left is an elution pool that is kind of broad. So you are less concentrated, which means you're going to use more buffers and your cycle times are going to go up. However, for jetted technology, since that uniformity coefficient in the distribution of those beads are much tighter, you have a much tighter elution pool. You also have improved resolution because molecules are exiting the column at similar rates. You're using less buffer volumes and your cycling is going to become shorter. Other advantages to the jetted technology really lie with the fewer small particles. The fewer small particles reduce your propensity for fouling, and also that leads to higher flow rates in certain cases because you don't have as many foul particles at the top of your column. With that, I'd like to move away from our technology and focus a bit on our latest product here, which is Jetted A50 High pH. As I mentioned earlier at the beginning of this presentation, Jetted A50 High pH is our newest product, and it's truly unique in the sense that there is no other Protein A product like it on the market. However, we did not want to just create a product for the sake of creating a product. We wanted to bring a product to market that our customers would value. We wanted to create a product where we met their expectations, whether it be capacity, specificity, stability. And if you look at the landscape of purification, there's a change in FC-based therapies. We're moving away from these standard construct MABs. We're having a lot more of these variants, whether they be fragments, multi-specifics, or FC fusions. And we wanted to create a high capacity resin that could allow for the elution of some of these more acid labile constructs at higher pH. So Jetted A50 High pH is able to elute up to pH 5. How we went about designing this ligand was with Repligen. We partnered very strongly with them. And through Navigo, a ligand company based in Germany, we were able to completely design a new ligand from scratch. And if you look at Protein A, naturally, you have 5 similar but not identical antibody binding domains, A, B, C, D, and E. Different vendors take different approaches to developing these domains. And what we did was we looked at all 5 of these domains. And through site-specific mutagenesis, we were able to create these different variants. And these variants were designed around parameters at the bottom right of your screen: sodium hydroxide stability, specificity, a higher elution pH up to pH 5, as well as high capacity. We designed several variants, which were then prototypes, and we selected a final variant. And through that, we multimerized and found a dimer to be the optimal configuration for the NGL Impact A High pH ligand. So how does Jetted A50 High pH perform? If you look at this graph here, we look at 2 different bispecific antibodies, and you can see Jetted A50 High pH in green. Our Praesto Jetted A50, our traditional Protein A, is in blue, and a competitor Protein A is in red. Jetted A50 falls somewhere in between. And what you see here is we have a rapid uptake in binding capacity and a fast plateau. That's really an improved mass transfer kinetic resulting from our jetted manufacturing technology. Jetted A50 High pH, as the name implies, is based on a 50 micron base matrix and has a capacity to bind up to 60 grams per liter. This is truly a pure FC binder with minimal VH3 interaction, unlike Jetted A50, which has FC binding as well as interaction and avidity for the VH3 region of an antibody. What you're looking at on your screen is a study that was done with Biogen in collaboration with Repligen and presented at ACS in the fall of 2021. On the left side, you will find an FC fusion protein. And through a linear pH gradient on the elution going from pH 7 to 3, we compare 2 prototypes of our high pH ligand to both Jetted A50 and a competitor Protein A. And what you find is the high pH ligand allows for the elution of this FC fusion protein between pH 4.8 and 5.2. However, traditional Protein A requires a more acidic condition for elution. So that's why you see the elution coming off around 3.4, 3.5. And this really demonstrates the efficacy of our ligand for what it was designed for. On the right side of your screen, you'll find 5 different MABs that were tested. Important to note, MABs 2, 3, and 4 are derived from the VH3 germline, whereas MABs 1 and 5 are not. And when we looked at the two high pH ligands that you see in blue and light blue, you'll find that high pH 001, the one in dark blue, required a lower elution pH for MABs 2, 3, and 4 in comparison with the high pH 002 ligand. And as mentioned, MABs 2, 3, and 4 are derived from the VH3 germline. As such, we found that the high pH 1 ligand had avidity for the VH3 region, and that really inhibited us from eluting at a higher pH. So we decided to move forward with the high pH 002 ligand, which is a pure Fc binder with minimal VH3 interaction. Now, I'd like to hand over the presentation to one of our customers, Kayla Barry, whom I've introduced at the beginning of this presentation. She's going to walk you through a case study and application of Jetted 50 high pH for the purification of bispecific antibodies at MabPlex, based in San Diego. Thank you.
Hi, my name is Kayla Barry. I'm a senior engineer in downstream process at MabPlex USA. The interest in Jetted 50 high pH resin came from our past challenges with bispecific purity, especially starting off over Pro-A resins. So MabPlex USA has assessed manufacturability for several bispecifics, bispecific antibodies or BSABs. BSABs can become really unstable under typical protein elution conditions, typically from pH 3 to 3.5. These conditions typically cause aggregation and fragmentation. The first molecule that we investigated, which is designated BSABZ, had major issues with aggregation and fragmentation. That made it very difficult to purify. You typically, after the Pro-A step, we only had about 60% monomer remaining due to those typical elution conditions. So as an example, this is a typical isocratic elution off of a usual Pro-A resin. You can see a pretty tight isocratic peak, but overall, looking at the graph in the bottom left, you can see monomeric purity starts off very low and only achieves about 80%. And this is about after a lot of development in the isocratic method. Cumulative monomer is only about 65. And you can see the high molecular weight species at the beginning of the elution start off about 40%. So there is a lot of, species that are undesirable here and almost a very poor separation of the monomer from the high molecular weight species. So this, due to such low monomeric purity off of the Pro-A step, it required further low yield chromatography steps in order to meet product specifications. So overall, throughout the whole process, we're not retaining that much monomer or overall product, and you end up with, very little product at the end of your entire purification run. So our interest in Purolite, Praesto Jetted 50 began, our high pH, Jetted 50 high pH, began when, we were introduced to this new novel resin, which could elute antibodies up to pH 5. So this was of interest to us because we were hoping that maybe our more acid-reliable bispecifics would be able to not crumple during the elution conditions. So BSABZ, HCCF is purified over a small-scale high pH column to evaluate elution at a higher pH to both prevent aggregation and or separate the undesirable species. So we wanted to see if this would have any effect. So for the Jetted 50 high pH gradient capture for BSABZ, we had a 3.42 column that we packed in-house. The low density was about 20 mgs antibody per mil packed resin, and we collected in half CV elution fractions. There was an earlier wash fraction. You can see that first peak after the load that was also collected, which is mostly low molecular weight species that were able to be separated just from a high salt wash. And then, we collected a gradient between pH 5 to pH, I think 3.5. And we were able to recover about 96% cumulative monomer and only saw that generation of high molecular weight at the very end towards the lower pH range, which seemed promising for the idea, supporting the idea that, eluting at a lower pH is what was driving further aggregation. So due to a lack of material of the BSABZ, we had to look at a few of our other similarly designed bispecifics that we had available and evaluate those. So we were looking at two other molecules that were similar, BSABX, which is mostly stable with some aggregation fragmentation, and BSABY, in which aggregation and fragmentation were very typical. And the only analysis we had for this was SEC. So obviously it would be interesting to look at how you're removing, host cell contaminants and, what other, how it's changing maybe the ICE profile or the CESDS. But for this purpose, we only had SEC for analysis. So high molecular weight species are definitely a major concern because those are the hardest to remove. Low molecular weight species are a lesser concern because those can usually be removed with an optimized wash step. But in this case, both molecules are better behaved than BSABZ. But BSABZ is sort of the one that sparked our interest because it was the most difficult molecule to purify. So our overall method, we used standardized 5 mil pre-packed columns of the Jetted 50 high pH and standard Pro-A. The low density was kept at 20 mg antibody per mil packed resin. And we collected in third CV fractions from 1,000 to 1,000 milli per centimeter of a path length, but normalized to a 0.2 centimeter path length, obviously. Gradient elution was from pH 5.8 to 3.3, 0 to 100%, the B pump on an active on over 20 CVs. So pretty typical the Pro-A method, gradient method. So for BSABX, the elution started at about 71% B, so closer to the lower end of the pH range. And you can see some separation visible in the UV trace. So for everybody's reference, the blue UV trace is referencing the Jetted 850 high pH run, and it's overlaid with the standard Pro-A run, which is in red. So you can see from the standard Pro-A run, the main elution begins at 86% B, and we get a much tighter peak with almost like no visible separation. So in terms of looking at our SEC monomer, our results, the Praesto Jetted 850 high pH had an overall cumulative monomer 90%, with the highest fraction being about 93%. And most of the low molecular weight species eluted first. We have a pretty standard high molecular weight species hanging out across the board for both of these runs. And looking at the standard Pro-A graph, the overall cumulative monomer is only 84%. So it dropped about 6% and no fraction was above 87. So you're not even getting close to within a typical spec of greater than 95% monomeric purity. And so pretty much all species on the standard Pro-A are co-eluting. And at least with the Jetted 850 high pH in this one, you can see that there's separation that you can, probably optimize in an isocratic method and get a higher, monomeric purity right off the bat from Pro-A. For BSABY, species separation follows a kind of similar trend to BSABX with greater separation from the high pH than compared to its BSABX, but you can really see that there's about two main peaks that are coming off during that gradient. And the elution begins much earlier, about 38% B, so at a much higher pH. And the separation is very visible. For the standard Pro-A, the main elution begins at about 86% B. And you can see it's still a tight peak, but there's a little bit more separation in the UV trace than compared to X. So you are getting a little bit more of a broadening of that peak, but it's still pretty tight to the end of that lower range of the pH. For the fraction results, BSABY had overall cumulative monomer of 96, which is very high, and a slight increase of the high molecular weight species at the more acidic pH as kind of expected. But overall, it was very high, consistent. For the standard Pro-A, the overall cumulative monomer is about 89%. But you can really see towards the end of that peak, there's a huge drive for high molecular weight species up to about 30% at the very end. Of course, those samples are going to be a little bit less in concentration, but it's still pretty consistent. And to have a drop from 96 to 89, where one resin puts your pool in spec and one takes, is far from it, is pretty surprising. And you can see that, there's co-elution as well, and you just don't get nearly as good separation from a standard Pro A resin than you would than you see the high pH does. So overall, the Praesto Jet A50 high pH elution conditions tend to result in higher monomeric purity versus a standard Pro A elution condition, I suppose. Elution with a standard Pro A resin at a lower pH looks like it drives aggregation and causes co-elution, which is, going to cause you a lot more problems down the line when you have to use a lot of other steps in order to actually separate the high molecular weight from the monomer. The higher pH elution functionality of the Praesto Jet A50 high pH resin just demonstrates overall separation recovery and might even put you, within spec for some of these molecules that it's hard to tell if they're coming out of the bioreactors, high monomer, and then just due to Pro A are kind of crumpling, but it kind of seems that way when you're getting overall good recovery, but half of what you're recovering is high molecular weight or low molecular weight species. So these attributes are ideal for platform capture for molecules that are very susceptible to this kind of behavior. And there's a lot more aspects of this resin, and I want to compare to my historical data to see, in the future I want to explore, are we clearing host cell contaminants? Are we damaging the molecule in other ways other than SEC? But this is a good first step towards resolving some of the issues we've had in the past. I'd like now to hand it over to Megan for the Q&A and answer any questions you might have. Thank you.